I'm very glad to be here to talk about science for peas. In fact, I put my pea plants outside today in our climate-warmed weather, and so I thought you'd like to hear all about genomics for peas. That's a pea weevil on a pea. And in Australia, this is a major pest of pea crops. And so, as is commonly the case these days, somebody said, well, I can figure out how to make a GMO pea that will make the pea weevil keel over and die. So they looked, it was associated with the Scientific Research Office of Australia. They said, well, these weevils don't bother beans and peas are closely related. What is it that beans have that the peas don't? And they found that there's an enzyme in the beans that interferes with the animal's ability to break down starches. And so the animal can't get nutrition from the starch in the seed. And so they produced a genetically modified pea where all they'd moved was this one enzyme from a bean to a pea. And that's about as close as it gets in genomics. You know, it's between very closely related species, as things go. It's not like from a Brazil nut into corn or something like that. And they thought, great, we're going to have this solved. Interestingly, because they were in a scientific rather than an agricultural department, the testing that was done was a little more thorough than usual. And in the course of that testing, they found that there was a slightly different modifying group being put on the product of this gene in a pea than there would be in a bean. And when they fed those peas as pea meal to lab mice, the lab mice developed an allergy, partial allergy, to that slightly modified group, which they wouldn't have done if they'd been fed bean meal because of that slight modification. And at that point, they didn't have a lot of funding. This wasn't a big private company. They just said, well, forget it, and they canned it. This is not typical of what happens to genomics projects, but this issue of is the food safe when it's been genetically modified is something that you've probably heard about because lots of different attempts at genetically modifying crops have been made. And one of the areas of major debate out there is the safety testing for the food produced by the genetically modified crop good enough? So here's an example where it was good enough, they detected a problem and they stopped. This is unusual. This is not what usually happens. And I know you've got some members who talk to you about things like this, and you've probably already heard stories about, you know, the Brazil nut protein, which was being expressed in a different thing, and what if somebody had an allergy to nuts? And there was a case where genetically modified potatoes were tested, and they proved to have some ill effects. Now, the problem is that very often there's great big political and business interests behind these crops. Usually it's multi-billion-dollar companies that develop them. And a number of the people who went out to prove scientifically whether or not this was safe and found that it wasn't safe, then ended up losing their jobs because there's a big push by the companies and the agribusiness-friendly politicians to get this stuff out, and they don't like pipsqueak scientists getting in their way. So if Phil Regal regains his health, you can hear some wonderful stories of that. Not wonderful in the sense that they're happy stories, but wonderful in the sense that they tell you just how deeply embedded the business interests are inside of the supposedly neutral agri-science parts of our regulatory systems. So the first connection to security comes for science for peas. If we talk about the security of our food supply, We can either talk about it in the positive way that Monsanto et al. Would like to say, okay, GMO crops are going to help improve the security of our food supply because these pests won't wipe them out and we'll get more even yields. Or they could make our food supply insecure in the sense that we might be getting foods that are not good for us. So that's one way that I connect security to genomics. Now, genomics started off as super high-tech stuff some years ago, but soon everything that anyone wants to will be sequenced. It's very exciting for scientists like me because we're at the point where that hasn't quite happened and we can get lots of new information out. But I was visiting a friend's house and I saw DNA SPA, DNA sequenced pale ale. And there's a depiction of part of the genome sequence of brewer's yeast on the side of the can. So that doesn't mean that this was genetically modified yeast. It just means that they used the tools of genomics to sequence the yeast. And in fact, I know a number of people who studied genomics and went on to become brewers. In fact, there was one guy who produced what he was sure would be a better yeast, did his own home batches of beer, and showed them to a microbrewery, and they said, "You're hired." So this serves to introduce the fact that genomics is easiest in microorganisms, things like bacteria, yeast, and viruses. And that's where it's gone the furthest. So we really have to think, if you think about genomics, yeah, there are genetically modified peas and so on, but almost all of the commercial genomics that's going on is happening at the level of a yeast, a bacterium, an alga, that kind of thing. And that's not in most people's visibility range. You're not thinking about that. If you asked where a lot of the money was being spent, it's being spent to create industrial strains of microorganisms that can, say, catalyze some chemical process more cheaply than traditional chemical methods would do. And so we've got a lot of modification going out there for the chemical, for the feedstock. There are people who are trying to make fuel for cars out of biomass. There are all kinds of ways that people are doing this, and usually they're playing with bacteria, yeasts, fungi. So when you think about genomics, think about this can of pale ale. Another absurd example that goes right along with science for peas, here's a genetically modified fish, the glowfish, trademarked, which you can buy and put in your home aquarium. It's made so that if you put a UV light on top of the tank, they glow in different fluorescent colors. So you probably can't read all this, but this is all the fine print that comes when you buy your glowfish, telling you that this is a patented variety, that you can't, that it's trademarked, you can't use the word glowfish in any way than other to refer to them, and that here's all the list of patents, and by the way, you absolutely must not breed these glowfish yourself and give them to other people. So this allows me, in a rather absurd context, to introduce the idea of intellectual property and control of genetic resources. And those of you who've been interested in this area for some time have probably heard many of the sad stories about farmers who were growing ordinary crops next to a field of genetically modified wheat, corn, soy. And then the company that owned the patent on the genetically modified crop came and said, "You're infringing on our patent, we're going to sue your pants off." Why? Because some pollen from their field had gone over to the other farmer's field and he saved his seeds. But he was now using their genetically modified intellectual property because some pollen crossed the road. And it sounds absurd, but there are people who've lost their livelihood or come very close to it because the companies have a multi-billion-dollar stake in this, and they will let loose a phalanx of lawyers on a farmer who they choose as a test case. So they're basically trying to keep all farmers in the camp of having to buy their seeds from the companies and not being able to keep their own seeds. But if you're an organic farmer and somebody across the road from you plants these things, we've come upon this recently, that shipments of soy from Canada can't be sent to Europe in many cases, because even if the farmer has not planted genetically modified soy, they can't prove that they didn't get pollen from genetically modified soy in, and so the Europeans won't accept it. So it's a big issue, and it all revolves around patents, intellectual property, and big money. So when we talk about security, we need to talk about the security of farmers' incomes and ways of life, as well as the security of our food chain. And here we have intentional breeding is strictly prohibited. So, of course, what did I do? I naturally Googled for glowfish. Well, the first thing that came up was one of their patents. And then the thing that came up was Kyla fishy, who's 14 years old just a few days ago. She just received a 10-gallon tank with a bunch of glowfish. She said, can I breed different colors of them? And so, here's a 14-year-old receiving some genetically modified fish, and her first instinct is to go out and breed them. And somebody answers saying, well, unfortunately, most of them are sterile. They've been treated so they won't breed. This is because they do breed true. And then there's a million people exchanging comments about that from the fish fanciers guild. Some saying, no, I've been able to grow them. And others saying, well, if you have, it's illegal. And yet it's a ridiculous example, glow in the dark fish. But Kyla fishy, if she gets bored with her aquarium, as many people do after a while, what will she do with them? Often they're not willing to safely dispose of them. And so they'll find either the toilet or a nearby pond and dump their fish in there. Now in Canada, I'm not too worried about the glowfish because they're zebrafish that have been genetically modified, and those won't survive a winter. But what if this was in Florida or some tropical area where zebrafish can thrive? So now you'll get genetically modified organisms being reintroduced in an uncontrolled way because 14-year-olds got bored with their aquarium into the environment. So that's where we begin to talk about security and ecology. I don't think some other fish getting a glow-in-the-dark gene is going to do bad things to our ecology. I start with the fun and rather foolish examples, but they introduce the general questions pretty nicely. Here's another fun and not so foolish thing you can do. This is from Carolina Biological Supply. Lots and lots of high school teachers buy the stuff for their science biology classes in high school from this company. And item number 211,086 you can buy for $62, and it's a glow-in-the-dark transformation teacher demo kit with prepaid coupon. Yes, I love that prepaid coupon. And that prepaid coupon is so that you can order more of the ingredients when you run out. Well, what is it? It's a kit that allows you in the classroom to genetically transform bacteria so that they will end up with genes that cause them to glow in the dark. Okay, so who's doing this? High school students, because a good teacher wants to have a hands-on learning experience. And an essential part of the process is that you don't get 100% conversion of the bacteria when you put in this little genetically engineered virus that goes into the bacterial genome and adds its stuff. So how do you keep only the ones that have been transformed? Well, the plasmid that the virus is inserting contains a gene for glowing in the dark, but it also contains a gene that makes that bacterium resistant to the antibiotic ampicillin. So then you treat your culture with this antibiotic, and only the ones that survive are there growing a few days later, and they're the glow-in-the-dark ones. So you've taught the students very nicely how this is done, and that kind of antibiotic selection technique is indeed commonly used. Where do you think those Petri dishes go when the class is done? Do they go into an incinerator or an autoclave, which is the safe disposal method? Or do they go into the school trash or get flushed down the sink? So $62, you can do it at your high school. You can do it at home. I'm going to get to that in a minute. But I'm trying to make the point that genetic engineering is not something that people in white lab coats do. It's something that people in schools do. People can get genetically engineered organisms, for their fish tank, in their beer, etc. So it's a very widely spread thing now, which is different from when we were first talking about it a few decades ago. Well, what is the bacteria they're transforming? Well, it's good old E. coli. And so this is from their text. The bacteria transformation takes up this plasmid, which is like a chunk of bacterial genetics, and makes it ampicillin resistant. Well, where does E. coli come from?
Gut. Intestines.
Yeah, poop, shit. It is the most common bacterium in human intestines. And what can you think of as residents of Ontario about E. coli?
Walkerton.
The E. coli, which killed a number of people and seriously damaged a number of others, especially children, was an E. coli which had acquired a dysentery toxin gene via a plasmid. So the toxin gene originally came from a completely different species of bacteria, but a plasmid, the same thing that we're giving our high school students to transfer the glow-in-the-dark gene, allowed this toxin gene to be transferred to this one particular O157:H7 strain of E. coli. Cattle aren't bothered by this. They don't have the specific receptor that Shigella toxin attacks. So cattle don't get sick with it. So cattle often harbor this strain of E. coli. And when the cow poop washed into the municipal wells, lots of people got exposed. And in fact, many of these bacteria were antibiotic resistant. And so giving the poor kids antibiotics actually worsened their condition by knocking out other bacteria and allowing the toxic ones to thrive. So a little bit interesting. So the general topic of this is when you talk to doctors, they talk about superbugs, that we are, by using a lot of antibiotics in various environments, we're making it advantageous for bacteria that get a plasmid containing an antibiotic resistance gene. I don't think it's advantageous that they get a glow-in-the-dark gene, but it's certainly advantageous if they get an antibiotic resistance gene. Well, where does this happen most?
Countryside?
Countryside, in farms is one place. Hospitals is another major place. So just think of where antibiotics are used the most. And they're used the most in farms and in hospitals. And not 100% broken down by municipal sewage treatment programs either. So sewage lagoons are a third place. So you can close your eyes if you don't want to see one of the results of getting a nasty thing. This, it's hard to see with these lights, but this is a really, really nasty infection on the hand of somebody. And it's caused by MRSA, which is one of the superbugs where if you get it in hospital, you're going to get real, real sick and you may die because it's already resistant to almost all antibiotics. Up until 2 or 3 years ago, there was still one antibiotic of last resort where if they knew that you had this, they could give that to you. It's now been reported that there's an MRSA in Thailand that's resistant to that antibiotic. So once that hits the hospitals, there'll be cases of people dying from bacterial disease, just like they did prior to 1920, because we won't have any drugs left to treat them with. And how is this happening? It's not happening because people deliberately set out to create antibiotic-resistant bacteria. They simply created the ecological conditions in which the natural genetic engineering that bacteria do all the time favored antibiotic-resistant bacteria. So we're doing genetic engineering without knowing about it, without thinking about it all the time, and that's a bit of an issue. So genetic transfer has been going on via natural vectors. They might be viruses, parasites, or even symbiosis for billions of years. We exist because of symbiosis between several types of bacteria that started off about 3 billion years ago. It's sped up enormously in hospitals and children's sickbeds due to overuse of antibiotics. And this is something our politicians and our medical colleges are really behind on. They all know that they need to regulate this, but we wouldn't want to tell doctors what to do, would we? We would give them nice messages asking them to prescribe less. But there are countries where there have been monitoring boards put in place and very strict rules about not giving any more antibiotics than you absolutely need. It's all true. The weight gain due to antibiotic treatment has been the big thing pushing their use in agriculture. And it needs to be legislated out. It can't be put into feeds because right now there are lots of animals where their basic feed is laced with antibiotics to achieve these weight gain. So antibiotic use in farm animals produces superbugs. And what do you think those ampicillin-resistant glowing E. coli are doing after they're disposed of by the class? They're just going somewhere else and passing their genes along. So here's a reference to the case. 4 people in Singapore have been affected by the new Delhi strain of something that combines all these previous antibiotic resistances with resistance to the last class of antibiotics that we had to treat them. So this is happening right now. People are getting very sick and in some cases dying in hospitals in Asia. The typical spread of these things takes anywhere from months to at most a few years. So it'll be in Canadian hospitals soon. And of course, the sicker people are with bacterial disease, the more antibiotics they get, so the more likely they are to accidentally create a proving ground for these kinds of superbugs. Well, we can go from the lab to the classroom to the art gallery. So in the year 2000, Eduardo Kac, who's an artist and professor, collaborated with French scientists to produce Alba, a white rabbit. And if you see Alba in white light, she just looks like an ordinary white rabbit. But if you put her in black light under UV, she glows green, because that same jellyfish gene that causes green fluorescence under UV light was transferred genetically into her. And there's a long address there, but if you Google Alba green glowing rabbit, you'll find it pretty quick. Now, artists who've been doing this are not necessarily doing this just because it's cool to be able to do this. And this is why, if you're appalled or upset by this, I suggest that you do Google it and follow up on the thing, because the artist statement said he wanted to make people aware of what was possible, and this was probably the best way he could find. And boy, did it work. He got newspaper headlines all around the world. He wasn't there to promote genetic engineering. He was promoting people thinking about it. And I'm going to come up with another example of that in a moment. But the fact that artists who are often in the vanguard of change have picked this up has led to some interesting unintended consequences. Here's a silly example. This is an artwork by Nathan Shaner in the lab of Roger Sien. You can see that it's sort of this beach scene. Each of those different colored streaks are a different strain of genetically engineered bacteria that glow a different color in UV light. So this stuff is getting out into the artistic community. People are playing with it for fun and to make statements. And this whole process of taking bacteria and changing them now has a name. It's called biohacking. And people treat it the way some of the older of us in the room might remember getting a radio kit and putting it together so you can say you built your own radio or electronic device. Or if you're 20 years younger than that, you might have built your own computer. Or if you're 20 years younger than that, you might have printed your own solid-state device with a 3D printer. So it's out there. And this book, Biohacking Gentechnik aus der Garage, Genetic Techniques from the Garage, was published by a set of 3 German journalists who also had some biology background. And they went out to see just how easily they could do this a couple years ago. And BBC carefully protects itself against screenshots. So I couldn't put the screenshots, but there's a very nice 3-part BBC series done by these people in collaboration with the BBC that you can find if you do BBC biohacking and look for that. And you can get all the details. They show how easy it was. They show how relatively cheap it was to get a fully outfitted genetic engineering lab in their garage. Actually, they used somebody's back room, I think. And although they did not actually complete it, they were able to order the gene for producing ricin toxin from a company. Now, ricin toxin is extremely lethal. And so the fact that they ordered this and that, it got delivered and no one came and broke down their door and arrested them, they cite as one of the facts that, if this is in the hand of somebody who's well-meaning and just trying to have fun, fine. But you could genetically engineer organisms that produce very lethal toxins. And all the stuff, they spent about $3,000 or $4,000, but they said to do less elaborate stuff you could get away with, maybe the cost of an iPhone to set up your own little lab. So biohacking is out there. And guess what? Now we're back to security again. Remember September 11th, 2001, and the packets of white stuff that started showing up, and the FBI freaked out because they feared that it was anthrax, and some of it was, but most of it wasn't. So the FBI actually has a well-buried department of bioterrorism. And these people are exactly as smart as most of the rest of the FBI, which is to say they have college degrees, but they don't always think or use them. Here's a FBI special agent for bioterrorism. And so they've adopted a soft approach for right now, which is where they go to hackathons, conferences where these biohackers get together and compare what they've done, and vendors come and show them the neat stuff they got. And they run little awareness sessions, and they try and tell people, well, look, we're not trying to stop you having your fun, but a bad guy could do this. And how would you know if a bad guy was doing this? So why don't you do, and they're trying to get people to think about the ethics of this and about the safety of this. This is the public face of what they do. Okay? Nice, public, trying to raise awareness face. So 2004, another artist, a professor at SUNY Buffalo, had an unusual experience.
Time of terror. Even Kafka might have had trouble conjuring up this one. Steve Kurtz is a professor of visual studies at SUNY Buffalo, and that's State University of New York, Buffalo, a founding member of the award-winning art and theater group Critical Art Ensemble. On May 11, 2004, his wife Hope tragically died in her sleep. When he called 911 for help, a nightmare that would last for the next 4 years began to unfold. The police became suspicious of his art supplies and harmless bacteria cultures that he was using for an anti-war project about the public health impact of germ warfare programs. Kurtz was detained as a suspected bioterrorist, his home raided by the FBI, the Joint Terrorism Task Force, Homeland Security, his belongings, his cat, even his wife's body were seized. After a federal grand jury refused to charge Kurtz with bioterrorism, Kurtz and his colleague Robert Farrell of the University of Pittsburgh Graduate School of Public Health were charged with federal mail and wire fraud concerning the acquisition of $256 of harmless bacteria. Under the Patriot Act, they could have faced up to 20 years in prison. After 4 harrowing years, on April 21st, a federal judge dismissed the government's entire
I'm not an expert at video editing, but the case was dismissed, but Kurtz was really lucky. he was a tenured member of a university where the contract for tenured faculty included the university supplying lawyers and support for people who were attacked, on their opinions that related to their work. And this is stuff that he had been exhibiting and had been in, he had done nothing wrong, but the FBI just went totally apeshit over this, totally. So if he hadn't had those resources, the results could have been quite different. If it had been one of these 17-year-old biohackers, think of what might have happened. So some people who've looked at this case think that the FBI wasn't really worried about Kurtz himself, but they just had a test case that they were trying to send a message of fear to the biohacking community.
The film, Strange Culture, that deals with the invitation to your exhibit. The invitation had some Arabic writing on it.
I believe they used the Arabic that Atlas Group used, which is a group that explores the history of Lebanon, that was on the cover of the exhibition announcement as enough proof to come to the opening and serve subpoenas. That is so wild that one could just use Arabic on any text in order to prove some sort of intent or ability to serve subpoenas. I had no idea that would be the use of that actual exhibition announcement.
That clip from Strange Culture, you can find out more about the film at strangeculture.net. Steve, we only have 2 more minutes, but he's talking about the invitation to your
Kurtz
art
there.
exhibit that was supposed to be at the Massachusetts Museum of Contemporary Art, the invitation they picked up in your house that day when your wife died.
Right. Yeah.
All right, so we'll get back to this in a minute, but what the speculation of the documentary makers and of a number of the friends of Steve Kurtz is that Kurtz and the scientists who had provided him with the bacteria were chosen as people who had been critical of government policy and or were prominent. And the thought was that if the FBI just hammered them with these big charges and got them convicted, that would send a big message of, don't you ever dare think about doing this or we will get you. So let's move from Steve Kurtz, who had all these resources and he was middle-aged, he had the energy, and he had a group of activist friends around him to support him in this. The scientist, the head of the Department of Genetics at the University of Pittsburgh Medical School, from whom he got the bacterial cultures, happened to have cancer at the time and was undergoing treatment for cancer when the FBI started suing him. The stress level on him got so great that he began to have a series of strokes while he was undergoing the chemotherapy. So in the end, he plea bargained for a lesser charge because he just couldn't stand it. He was also driven out of his department chairship. He didn't feel like being in the documentary himself, but he wrote out a statement which they got an actor to read. So these are the words of Professor Farrell, read by
growing body of evidence against the ways that the government, the military, science, and industry often collude against the public good. And this is primarily why he's being focused on. This is why he's seen as a good target.
An excerpt of the film Strange Clip. I want to turn, Strange Culture. I want to turn to another clip now. This features the actor Peter Coyote reading a statement of Robert Farrell, very well-respected geneticist, head of the Department of Genetics, former head of the Department of Genetics of the University of Pittsburgh Graduate School of Public Health.
From the beginning of the whole affair, I felt that I was simply collateral damage. From the start, the FBI seemed to have been interested in Steve's politics. I got the impression that they were against the activities of the art ensemble and Steve, and they were out. This was a way to get him. Steve has an idea that they want to frighten anyone, including scientists, who question what go on in this country. I don't think I did anything wrong. I would do the same thing again with Steve or anyone if they wanted reagents and they were serious people that convinced me that they were doing all this for educational reasons. That's the whole point, to educate the public about what is going on in science today. If I've broken any law as far as Steve's concerned, I've broken it 1,000 times because I've had many people in my lab to learn these techniques and given biological reagents to many students to work with. Genetically modified foods inserts genes from one kind of plant or animal into another, using viruses as a carrier. We have no idea what the long-term repercussions of these transplants are going to be. Genetically modified foods are not labeled. You can't go into a supermarket and find out that the milk you're drinking has bovine growth hormone in it, or that the tortillas you're buying are made with genetically modified corn, or that the soy you're buying has been made with genetically modified soy. So all the citizens of the United States have been turned into unwitting experimental victims of a mass marketing industrial phenomenon which has no regard whatsoever for their health, whose regard is basically about increasing wealth for shareholders and increasing the viability of the corporations and increasing the lock on the food supply of the corporations. The choice of whether to consume genetically modified crops is everyone's to make for themselves. The agribusiness has introduced these products into the food chain without people being aware of it. It has all happened with the government's tacit approval.
All right, so that clip from the actor reading the words of Dr. Robert Farrell gives you an idea of what a serious scientist who's thought carefully about the subject says about this. I'd like to make clear, I'm a geneticist. I've worked with genetically modified fruit flies. I have no objection to this as a scientific technique. But as with anything, once you move into stuff that can impact people, people don't eat fruit flies, thank goodness, and fruit flies are not a major pest in the field, you need to have certain safety procedures in place. And the tests of whether things meet those safety procedures needs to be done by independent groups. But here in Canada, in the United States, and in most other major industrial countries, the system that looks at food safety is heavily penetrated by the business that it regulates. And so the people who provide the supposedly independent judgment on the safety of these things very often are not working at arm's length from the people they regulate. And in fact, there's a really well-established career path where somebody works in one of those government agencies for a while at a kind of ordinary wage, and then they leave and they get hired for big bucks by one of the companies that they regulated. This is not unique to food organizations or GMOs. It happens everywhere where there's regulation. But it is an insidious kind of corruption of the regulatory system that goes very deep. And it goes into persecution of people who dare to publish results that go against what the companies want. We probably won't have time today to tell you about all the examples of that, but there are lots of examples, not just relating to genetically modified food, but relating to toxins, to pesticides, to a variety of other things, where there are well-documented cases of, let's say, a government scientist coming honestly up with the conclusion that, hey, this genetically modified crop didn't save farmers money, and he publishes a paper and he's fired from the government. Whereas just the year before, he was getting all these awards for being such a great researcher in the government. So tons of pressure is exerted. It's very hard for a lot of people to resist that pressure. So where are we so far? Gene transfer happens naturally. It's part of the whole system of life. We wouldn't exist without it, but amongst bacteria, it's happening at increasing rates because of our incautious use of antibiotics. We've seen that genetically modified organisms are sold as school and hobbyist kits. toys, as home aquarium fish. So it's pervasive now. It's not something that's still in the lab. Intellectual property, or IP as the venture capitalists love to call it, plays a very big role because once you put genes into an organism, well, organisms reproduce. How are you going to make sure that you, the one who created this genetically modified organism, still control that product? And so there's this whole set of layers of intellectual property law that have been built up around this. And it started off as we began sequencing genes with some really nasty cases, one of which you might have heard of. As soon as people found that there was a gene called BRCA2, where if you had a mutation of it, your odds of developing breast cancer were much higher, a company in Utah patented the sequence of the gene and their test for whether or not you had a mutant copy of this. Well, guess what? The price of the test got real high and stayed real high. Just recently, I don't know whether it was last year or the year before, a public interest group bringing together lots of people, but largely funded by breast cancer survivors, managed to win a court case invalidating that patent, which means that tests for that, whether or not you carry that gene, are now available much more cheaply. But this whole idea of intellectual property and genetics is a really fast-changing and difficult area with lots of money floating around. So one of the battlegrounds is our food supply. Another battleground is our hospitals. Another battleground is, the bioterrorism area. And I think it's legitimate to be worried about bioterrorism. But frankly, if I wanted to poison a bunch of people with ricin, I could order some castor beans and extract the ricin with a $10 home chemistry kit. I don't need a genetically modified bacterium to produce ricin toxin from castor beans. So sometimes the fears are legit, sometimes they're overused to put a scare into opponents of government. So we had ecology in the title. So let's switch to where genetically modified organisms and ecology might be something you'd be interested in. So this is a quote from Wikipedia. It says, "More recently, the law of unintended consequences has come to be used as an adage or idiomatic warning that an intervention in a complex system tends to create unanticipated and often undesirable outcomes." Common sense translation of all this, we know that much about how the world runs. So when we go and poke it, very often we can't predict what that's going to do. Ecologists are some of the most humble scientists I know because they know how much we don't know. So ecosystems are very complex things. So let's examine some concerns about ecological unintended consequences of genomics. And we're going to get, it'll be a little bit dry at first, and then we're going to get to some hot stuff. So here's a picture of monarch butterflies that I took about 15 years ago in a country plot that I had. And I'd planted a bunch of milkweeds there, and during the fall migration, I would end up with hundreds of monarchs in my yard. I had a collection of monarch wings that I collected from dead monarchs that I found beside the nearby highway, because in that season there would be so many that were hit by cars and dropped down dead. I could just pick up bags of them, and then the wings were very colorful. I could use them as decorations. So you've heard probably about the herbicide Roundup. Roundup, otherwise known as glyphosate, is a herbicide that was developed because it's much less harmful than DDT to the environment and to people. So as a first step, it was a good step. Monsanto, after they developed Roundup, was saying, well, this is great, we can nuke everything in a field, but if only we could make the crop resist it, this would be great. So they tried for 10 years to put resistance to Roundup into crop things, unsuccessfully. And then one of their employees noticed that a sump from their Roundup manufacturing process was getting clogged with green goop. Well, it ought to have been green because Roundup kills green things. So they took the slimes that were in there, took them back into the lab and said, look, they have a gene. And so by the same process by which we accidentally do genetic engineering by flooding the environment with antibiotics, Monsanto accidentally did genetic engineering by flooding the sump with a leftover Roundup. And after a while, something that resisted Roundup they then extracted that, transferred it into corn and soy and other crops. And now farmers can plant Roundup-ready corn, Roundup-ready soy. It's gotten to the point where when you go and look at a farm catalog, you have to know that RR or RY in the catalog name means that it's Roundup ready, because then they can go out and nuke the field with this herbicide, and their crop plant will resist it. Now, there's a whole controversy that I'm not going to talk about the toxicity of Roundup. Not going there because that's not genomics. But the unintended consequence of producing the genetically modified crops that resist Roundup means that the environment is now being doused in Roundup. If you traveled across southern Ontario 20 years ago and you looked at corn or wheat fields, not cornfields are too high, if you looked at wheat or soy fields, you would see lots of milkweed stems sticking up from them because milkweed, the most common variety of milkweed, is a perennial. And it's kind of hard for farmers to get rid of. It actually takes labor to get rid of. And farmers who've got, 1,000-acre or 10,000-acre farms don't want to pay for a lot of labor. How am I doing on time here?
You have 5 till 8. You can go another 15 minutes.
Okay, that'll do that. Then I'll tell you the story of how you got rid of milkweed in the old days. My best friend grew up on a farm near Guelph, Ontario. And there were 3 brothers, and Dad gave them like a nickel if they went out in the fields and came back with a bundle of milkweed stalks that big under their arm. And when you pull the stalks a couple of times, the plant eventually loses energy and won't regrow. So that was a herbicide-free milkweed control thing, but it involved labor. So now with Roundup-ready crops, there's no milkweeds in the fields. Well, the herbicide usage has gotten about a 60% decline in milkweeds in general because so much of the area in which monarchs live is agricultural. So the monarch population has been crashing. You probably heard about this. And the National Resources Defense Council filed a suit against the EPA last year in which it argued that the agency ignored warnings about the dangers of glyphosate Roundup usage for monarchs. So that suit is ongoing, but it's not the glyphosate usage itself, it's the fact that they had a genetically modified crop that resisted it that made the Roundup usage get so big. There's another thing that's got an Ontario link to it that maybe I'll get to in a moment, but let's just look at those are the total area of monarch colonies in the Mexican wintering grounds over years. So each one of these is a year. And it fluctuates year to year from things like weather. But you can see that there's been a general decline. This is the year when Roundup-ready crops were introduced. And of course, they weren't fully used by farmers for a couple of years afterwards, but you can see this is the amount of Roundup-ready crop with each year after this point. And so now it's to the point where most of Iowa, much of Minnesota, many of those central western states where corn is grown are Roundup-ready almost everywhere, which means that Roundup is being sprayed almost everywhere. This stuff in dark brown is usage of Roundup on agricultural land. So the dark brown are the areas where it's really heavily used. Now, what happens in the springtime? The monarchs have been down here at their overwintering grounds in Mexico, and they have to fly up and have several generations on milkweed before they get to Canada. Where are they having these several generations? In the areas where the heaviest Roundup use is being done. And so the knockdown of milkweed has been particularly intense in just the areas that the monarch northward migration needs milkweeds in to succeed. Now you'll still find milkweeds, but the numbers have been decreased. And there are other reasons for the monarch decline, like the cutting down of forests in Mexico and climate change. But if it hadn't been for that slime in the sump at Monsanto 30 or 40 years ago, monarchs might be in much better shape than they are now. So how's that for an unintended consequence? Oops. So that was one simple example. Let me take you now to an ecologically more intense example. This is a case of us accidentally wiping out a species. Monarchs are on threatened species lists, and they may be extinct in another decade. But that was accidental. What if we deliberately started driving species extinct? Most of our driving species extinct has been accidental and, just we don't think about it, we pave a few billion more acres and they don't have anywhere to live. But with the methods of modern genetic engineering, we can now target ways to drive a species extinct. So, should we? So before I put up any information, I just ask you to think about this. Think about your own feelings about this thing. So we have the ability to get rid of species. Can you think of a species that we got rid of deliberately?
Smallpox?
Exactly. Perfect answer, because that raises the, why would you want to get rid of a species? Well, if you think of all the misery that smallpox caused, and the fact that it only lived in the human reservoir, so once it was gone, that was a good thing to do. You had a...
I was going to use a similar example, except that now our organism, which is one of billions of types of organisms on the earth, is overgrowing out of control.
Yeah, okay.
Disease used to be one of the ways we kept human numbers down.
All right, well, I'm not going to advocate for making you all sick so that monarchs will survive. There could be a vigorous discussion about that. We're trying to wipe out polio, because polio is like smallpox, that it only lives in the human reservoir, and if only they would stop killing polio inoculators and inoculators in Islamic regions of the world, we would have gotten rid of it by now. But talk about unintended consequences. How did the CIA figure out for sure that Osama bin Laden was living in that compound in Pakistan? They got a local doctor who went around inoculating children for polio, and he sucked back a little bit of blood from Osama's kitties and sent it to the CIA, and they genotyped and said, that's him. Well, now people in some parts of the Islamic world think that anybody vaccinating for polio is an agent of the CIA. Talk about unintended consequences. But that's not genomics, so we'll move on here. Here's a current issue, quite an important current issue. This is from The Guardian from a few days ago. And you can find it in many other places, and people are publicly saying, should we wipe mosquitoes off the face of the earth? Now, if you go to read the third and the fourth and the fifth paragraph, they say, oh no, we're not targeting all mosquitoes. We're targeting the mosquitoes that spread malaria, that spread Zika virus, that spread dengue fever. And of course, we've been trying to wipe them out with pesticides and other methods for years. That's why in Canada, we don't have to worry about malaria and yellow fever, even though that used to happen here. And with climate change, many of the species that do transmit those things are moving up into North America, so the issue is becoming lively again. So, what do you think? Should we take something like Anopheles gambii and get rid of it? Here's the quote. Even the great and generous E.O. Wilson, author of the touchstone argument for preserving biodiversity in his book, makes an exception for this mosquito which spreads malaria in Africa. Keep their DNA for future research, he writes, and let them go. Because this mosquito only bites humans. It's become specialized on that. It's one of the primary vectors for malaria. And malaria kills more people, especially more children, than almost any other illness around the world. But because it's not a first world problem, we don't see that in our headlines every day. In Africa, relatively few children make it to the age of 5 in some areas. And one of the main reasons they die is malaria. So there's a real issue there. And we still haven't been able to make a malaria vaccine. The pesticides that we've been using against the mosquitoes are stopping working because the mosquitoes have been evolving resistance to them. Doctors who write about malaria, I just read an article today in a reputable journal, are quite worried because they don't know where the next generation of pesticides will come from for controlling the mosquitoes. So, ta-da! Genetic engineering. We'll just put some genes into mosquitoes that can spread as they mate and eventually make them all die. So, this isn't the phrase, this is an easy to understand phrase. I'm just calling it lethal genes. Now, they're all shades of the spectrum. There are genes that are not lethal but make the animal sterile. But that's lethal for the species, even though it's not lethal for that animal. So one of the species of this thing that's more of a problem in the Americas, Aedes aegypti, has been genetically modified to suppress its own species. And it was developed by Oxitec, a spin-out of Oxford University. Field trials, and this isn't something people are talking about, this has been done in field trials. They take this genetically modified mosquito out, let them loose in large numbers, and they've done this in the Cayman Islands, Brazil, and Panama, and they reduces the number of these blood-sucking mosquitoes by about 90%. Now, this particular variety is designed to need to be reapplied. It doesn't completely wipe the species out. And this represents a great money spinner for Oxitec because they have to keep growing new supplies every year. This is not a new strategy. There's a pest of livestock in the southern United States called screwworm. And for years and years and years, the U.S. Agricultural Department has been breeding sterile screwworm males and letting them loose in the environment to keep the population down. The modified mosquitoes were approved by the National Biosecurity Technical Commission for release throughout the country. And now the city of Piracicaba is leading the world's first municipal partnership releasing these genetically modified mosquitoes. Well, that's a technique where you have to keep applying them. But there are other techniques where the genes that you insert promote their own transmission, so they spread quicker than they ought to. And you can set them up so that they won't kill the animal if there's only one copy of the gene, but if they get 2 copies of the gene, they die. So what happens when you do something like that? It spreads quickly through the population because at first, most of the things that receive it only have 1 copy, and so they're not getting the negative effects, and the thing has got a way of making itself spread quicker through the population. But then once it starts reaching 50% of the population, many of the matings in the next generation have 2 copies and they die off. And that process can go to completion. In other words, you can destroy the species completely. So perhaps we should do that with humans and save the planet. Yeah. Unfortunately, the human generation time means it would take about 10,000 to 100,000 years to work properly.
Just a quick question. I read a counter-argument just a couple of days ago to the one stated up in the white rectangle that when it comes to mosquitoes, et cetera, because according to the counter-argument, they're a very important food source for avian life and other
right.
parts of the
and this is what I meant about the 4th and 5th paragraph. The only mosquitoes that this is being thought about are the ones that are specialists on human beings. There are thousands of species of mosquitoes, I'll get to you in just a second, And wiping out a few human specialist species really doesn't affect the aquatic systems
Yeah,
at
well,
all.
the title there is a little misleading
Yeah.
because you didn't.
Yes, your question.
How do we know like the lethal genes won't mutate to be lethal to other species? Or is that dependent on the technique?
Well, so it's a great question. I've actually got that question again in next slide. So the first thing we want to do is to make sure that these don't cross species barriers. So if you were going to be approving a technique like this, you'd want to have some very careful biology done to prove that there are reproductive barriers between the species you want to wipe out and any other non-target species. Having said that, I think it's a little safer with things like insects. Genes don't hop as easily from one insect to another as they do from one plant to another. So would certainly have to be looked at. The next question is, could this get into some other vector? Could it get into a bacterium or a virus, which could then transfer it? And if you did that, would you suddenly have this spreading cancer of the ecology where all kinds of species would start dropping dead? I think we need to think about that, but it's a great lead into some of my follow-on slides. Yes, you had a question.
I just have a quick question because I'm not familiar. Do the different species of mosquitoes interbreed usually?
Not usually, no. There are some interesting special cases. The species that attacks people in Africa looks like it evolved from a treetop species that mostly attacked monkeys. And in some parts of Tanzania, you can still find things that can interbreed with the human specialists, but that also bite monkeys. So there's a case where there's a fuzzy line on that. That's why I say you need to do the hard work before you jump off a cliff like this. But you'd be saving, 1 million children from a very painful death if you wiped out 4 mosquito species in the world. So it's not something that we're going to be able to come up with a flip, no, you should never do something like that, answer. And we would also eliminate vast amount of spraying of toxic substances into the environment. So it is not something with an easy answer. So this is just talking about the gene drive. I've already talked through it, so I'll just flip on to the questions that you've already been asking. Can they leak? Can these lethal genes leak or mutate? And then the next question is, how much unintended consequence, which you got at, is it like, is this animal or plant important in the ecosystem? And we're pretty sure that for human-specific mosquitoes, they're not very important in an ecosystem sense. But I'll give you another example in a moment where a similar transmitter of disease is very important. So these specialists have a low ecosystem disruption risk. But there are large chunks of Africa where people cannot keep cattle, and where in fact it's difficult for people themselves because the tsetse fly transmits sleeping sickness. Now, the impact on people has begun to drop because we now have drug treatments for sleeping sickness. This is Africa, so you can imagine that the poor are not getting those treatments as much as the rich. But it's reducing the impact on people. But in large parts of Africa, your wealth is measured in the number of cattle you own. And there are portions of Africa which just happen to be those areas that you tend to go on safari to, where people haven't moved in and taken over the ecosystem. Why? Because their cattle die of sleeping sickness because they don't have any resistance to it, and the tsetse fly bites them. The tsetse fly bites the zebras and it bites the elephants and so on, but they all have some resistance to it. So let's say we wanted to wipe out the tsetse fly so that people wouldn't get sleeping sickness anymore. We would suddenly change a major aspect of the African savanna ecosystem. So the ecosystem disruption risk from this kind of species extermination would be very high. And because it would act on many different species simultaneously, very hard to predict. So this is one that you wouldn't do without an enormous amount of thought and public discussion going into it. I know what would happen. I don't have to predict what would happen to the ecosystem. The moment the cattle could survive, people would flood in from surrounding areas. They would wipe out the remaining large herds of native animals. And so the biggest ecosystem risk would simply be that humans could move into that area. Now, there would be other changes to the ecosystem as well, but humans are the most dangerous animal. So my opinion is that these gene drive technologies will be used in the next 5 years. This is from the Federal Register of the Journal of the United States. Didn't put all the stuff in, but it gave this company the right to release genetically modified Asian tiger mosquitoes in 4 different states in the United States, beginning in 2013 and ending this year in an attempt to drive down their numbers. Well, the Asian tiger mosquito is one of those species that can transmit Zika. It's very common in the US South and goes up the East Coast almost all the way to New York. I will confidently predict to you that Zika will be in the United States this summer, spreading very rapidly because this and the other Aedes mosquito that's there will spread it quickly, and because it spreads by sex and maybe by kissing, who knows what else. There will be a major panic because it now appears from good scientific studies that are coming out that it doesn't matter which stage of pregnancy you're in. If you have no resistance to Zika, you get bitten by a mosquito and you're pregnant anywhere from the 1st month to the 9th month, the fetus can be badly, badly affected. So, just for those of you who like to think about political consequences, when will this start to spread? Well, it'll start to spread about mosquito time, which in the southernmost states is about now. When will it go red hot in the headlines? I don't know, but think about July and the Republican convention and all those people who hate abortion. I imagine we will see some extraordinarily interesting politics around the spread of the Zika virus. And one safe way out for all of them, without having to touch red-hot things like abortion, is to say, get rid of the mosquitoes. And so if you go out and you look at municipal pest control agencies, they're all stocking up on pesticides big time because they know that they're going to be citizens screaming at them to get rid of the mosquitoes. And cities that previously had very low pesticide loads will be doused with them this summer. And so, the idea of wiping out the mosquitoes, the 2 mosquito species that transmit Zika in the United States, might be seen as less damaging to the environment than dousing much of the country in pesticides throughout the spring and summer months. So I would say that we really need scientific, political, and ethical leadership both to work through the process of how do you evaluate whether this is a thing that you can do, how to look at the potential unintended consequences, and we really can't afford, if we're going to flip a switch and wipe out a species, to have this corruption of the regulatory system. So we need to build something that is resistant to the powers that be. And these agreements need to be like the chemical weapons ban. They need to go around the world. They can't be on a country-by-country basis. So I've gone from glowing fish and funny beer cans to some things that I think are going to be very, very hot, very political, have to do with genetic technology. So now let me talk about all the stuff I haven't talked about, which is synthetic genomics. Most of what I've talked about revolves around transferring a naturally occurring gene from one organism to another. And that's the definition of genetic engineering. But there are people with much more ambitious goals than that. They would like to create completely new genes that never existed before, new modes of genetic transcription and so on, so new genetic codes, and new species. All of these are being actively done right now. So this is called synthetic genomics because rather than transferring natural genes, maybe with slight modifications from one place to another, You're creating completely new things that never existed before. And I will give you an example of one researcher who just helps tell the story really well because he's a great scientist, an arrogant asshole, and a huge promoter of this, and he's rich. It's not Donald Trump. It's J. Craig Venter. He is the father of the human genome. He actually got the genome sequenced faster than the US government. He's very bright and very impatient. And if someone doesn't go along with him, well, he just goes off and does it himself. He helped found a couple of companies, and he got some basic patents that have made him a rich man. And in an interview in 2007, they asked, assuming you can make synthetic bacteria, what will you do with them? And he said, over the next 20 years, synthetic genomics is going to become the standard for making anything. The chemical industry will depend on it. Hopefully a large part of the energy industry will depend on it. We really need to find an alternative to taking carbon out of the ground, burning it, and putting it in the atmosphere. That is the single biggest contribution I could make. Now, this guy is highly effective, very bright, rich, and he doesn't pay attention to namby-pamby things like rules and regulations. Yeah, he claims to, but he usually finds a way around them. So he started a company called Synthetic Genomics, which is partly owned by J. Craig Venter. And they do lots and lots of stuff. A lot of this is in conjunction with the chemical industry, that whole idea of, if you can make the great huge chemical pipeline shorter and use less weird stuff in it by putting some genetically modified organisms in. There's lots of that stuff going on. But this is one that I thought might raise a few hackles around here and get some interesting discussion going. This is a quote from Venter. One of the most important programs at Synthetic Genomics is the company's collaboration with United Therapeutics, in which it is literally rewriting the pig genome to create pig organs that will survive in humans as replacement organs for transplantation, e.g. heart, lungs, kidneys, and livers. Now, there are probably people in the room who've had a friend or relative who desperately needed a replacement kidney or lung, and they're very hard to get. And a lot of people die while waiting on the transplant list. So this is not a trivial social problem that he's talking about here. It's a life and death matter to many people. And the biggest problem with receiving a transplanted organ is that usually, unless the person that it came from is very close to you on a number of different genes that define immunogenicity of tissues, your body will reject the organ. And so you'll go through all this agony and pain, and then 6 months later the organ will die and you'll be sicker than you were before. So pigs, strangely enough, although they're not closely related to us, in many respects, their tissues are not that dissimilar from our own, but they have a whole bunch of genes which our bodies recognize as, oh, that's pig protein, let's get rid of it. So this is an idea to create genetically modified pigs that express none of the pig proteins that the human immune system recognizes. Because then when you transplanted the heart or the lung or the kidney into the transplant recipient, their body wouldn't reject it. Species. So there are other issues around this, but that's one of those unintended consequences that people don't think about. Now, do you know what's a great example of pigs acting as a reservoir of viruses for humans? The flu is the answer, yes. The flu is a native virus of bird populations. Birds migrate. In portions of Asia, farming styles for small farmers usually involve a pond, often with ducks in it, and a few pigs. The pigs and the pond aren't necessarily separated. The ducks catch the virus from migratory birds. most of the time it can't infect the pigs, but you get thousands and thousands, maybe even millions of exposures of the pigs to the ducks that have the flu. After a while, a mutant arises that can move into pigs. And now that mutant has adapted to an animal that's much closer to humans, and that's how we get a lot of the flu strains that eventually end up infecting us. So that's another example of unintended consequences and the complexity of the ecosystems we live in. So Craig Venter says we're going to rewrite the gene, put a wholly new synthetic construct and landing pad in the pig genome. Other cases, maybe there's only minor edits and we can just convert the pig sequence into a human sequence. And so these are his words. It's from an interview published on March 1st of this year. This is happening. You know, whether they'll let him do this, he's still investing in it. He's got big money in it. I didn't put in the picture of the pigs glowing in the dark, but there is a glow-in-the-dark pig as well. And this is a picture. Fortunately, you can't see the details. In Korea and in China, people have taken pig organs and transplanted them, in this case, into a baboon, in another case, into a different primate, and they lived for a while. And that's a success, because it's sort of proof of concept. And in this picture, one of the things you can't easily see is the army epaulets on this one person in the operating room. So we know what happens in China particularly, where the army has a big stake in a number of things. So a lot of investments in new technology are often driven by and controlled by government organizations in environments like that. So just because we might not want pig organ plantation to happen here doesn't mean it won't happen somewhere else in the world. So that's the end of my talk, and I hope I gave you a flavor of what's happening here. I didn't go into all the ramifications of synthetic genomics. It would take a whole other talk to do that. But I think you can see that there are lots of ways that science is involved, lots of ways that security, interpreted in many different angles, is involved, and lots of ways that ecology is involved. Thank you.